Welcome to the first series of lecture on amino acids. What we are going to do here is to go through the slides slowly so that during the class we can quickly recap the lecture on amino acids. Now, at the end of all the lecture series on amino acid, you should be able to understand the general structure of amino acids. You should be able to classify amino acids on the basis of their side chain. You should be able to relate the structure of amino acids to their function, especially in relation to protein structure. And you should also be able to recognize uncommon amino acids of physiological relevance. What is not covered in the lecture series on amino acids is the acid-base properties of amino acids. Now, this particular section will be covered in the Foundations course by Professor Bayumi. So let us first start with a very simple idea. How do you define an amino acid? Now, in textbooks, there are various definitions of amino acids, but I like to define an amino acid as the monomeric unit of a protein. That is, the monomeric unit of a protein is defined as an amino acid. Now, let us take an example. So I have here taken an example of ribonuclease A, which is a protein that is present in our physiological environment. And if you take this particular thread-like structure shown here and then unfold it, that means the coils are unfolded, you will get a very signature sequence of amino acid shown here in this part of the slide. And in this sequence, the first amino acid is lysine, which is designated by the three-letter abbreviation of the amino acid. And the last amino acid is valine. So if I want you to read the sequence of amino acids in a protein, you start from here and then you continue and come till the end, which is valine. Also, the sequence of amino acid in a protein is the signature of a particular protein. That means the sequence of amino acids in a protein, in a specific protein, will always be the same. So if I tell you, is this protein a ribonuclease A? What you are going to do is to look at the sequence of amino acid. So here you have lysine, glutamic acid, threonine, alanine, 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 so forth and so on. So if you continue to read the sequence of ribonuclease A, every time the sequence will be the same. So the arrangement of amino acids in a specific protein in the physiological system is always the same. Now, also, the other aspect that I should mention here is that not only the sequence of amino acids, but also the number of amino acids in a specific protein will always be the same. That means if I look at the total number of amino acids in ribonuclease A, it will always be the same. So coming back to the definition, the monomeric unit of a protein is defined as an amino acid. We move on and we try to look at the general structure of an amino acid. Before we do that, sometimes people are asking how many amino acids are present in nature. Now, of course, there are 300 naturally occurring amino acids. Now, this is an arbitrary number. That means an approximate number. There are specific amino acids that are still being discovered today. But of these 300, we need to focus on only 20 of them, because these 20 amino acids constitute the monomer units of proteins. In your textbook that has been suggested, you will see a list of these 20 amino acids. I have uploaded a particular handout on the learning management system which shows these 20 amino acids. Now, on this part of the slide, we have the general structure of an amino acid. So you can see here each amino acid has a carbon atom, and we call this the alpha carbon. Now, what is the alpha carbon? If I want to define it in terms of organic chemistry, the alpha carbon is the carbon atom that is bonded to the functional group in an organic molecule. In amino acids, the alpha carbon is bonded to a carboxyl group, that is the COO minus group, an amino group, that is the NH3 plus group, and a hydrogen. So of the 4 arms of an amino acid, at all times the 3 arms will constitute of a carboxyl group, an amino group, and a hydrogen. The 4th arm is bonded to a specific chemical moiety which is defined as the side chain. So in an amino acid, what will provide its unique chemical property? The answer to this is the side chain, or the R group. So if I want to summarize what I said in the previous slide, one amino acid will differ from the other amino acid in terms of the side chain only. That means all amino acids will have a carboxyl group, an amino group, and a hydrogen. So of these 20 amino acids that I mentioned that you need to focus on, let us take 2 out of these 20 amino acids. I have taken 2 amino acids, that is glycine and alanine. Now let us look at the 3 arms first. So they have, both of them have a carboxyl group, both of them have an amino group, and then both of them have a hydrogen. But what is different? What is the difference between glycine and alanine is the side chain. In alanine, we have a CH3 or a methyl group, whereas in glycine, we have a hydrogen. So I want to emphasize this because this is very important for your assessment. How do you differentiate between amino acids? The answer to that is that we differentiate amino acids based on their R group, or to be very precise, based on the side chain. Now we need to look at a particular aspect of each amino acid. So what I will focus on is the R group. So I can say that the side chain or the R group confers the chemical signature to the amino acid. Again, of the 20 amino acids, I take 2 amino acids. One is lysine, the other one is glutamate. Now, again, in these 2 amino acids, both of them have a carboxyl group. So both lysine and glutamate have a carboxyl group. Both lysine and glutamate have an amino group. Both of them have a hydrogen. But what is unique for lysine and glutamate are their side chains. When I look at the side chain of lysine, I see that it is positively charged. Whereas when I look at the side chain of glutamate, I find that it is negatively charged. Or in other words, I can say the lysine molecule is a basic molecule because of the presence of this particular chemical moiety, whereas the glutamate molecule is an acidic molecule. What you can see here is that the difference in the chemistry of the side chain confers a unique chemical signature to the amino acid. Now you may be thinking why I'm focusing so much on the side chain, because during the assessment, when we ask you a specific question, the question is always related to the side chain. We generally do not focus on the other three chemical moieties whenever we ask a question during an exam. We move on. We have to understand a bit of the stereoisomerism in amino acids. Now, the whole concept of stereoisomerism and isomerism will be dealt in detail by Professor Bayumi in organic chemistry. But we need to know a bit about the stereoisochemistry of amino acids. So we go back to the general structure of an amino acid. We know that all amino acids have an alpha carbon. The alpha carbon in the amino acid is bonded to 4 different chemical groups. So we have the carboxyl group, we have the amino group, we have the side chain, and we have the hydrogen. So what I can say that all amino acids have a chiral center. What is a chiral center? A chiral center is an atom that has 4 different groups bonded to it in such a manner that it has a non-superimposable mirror image. I repeat, a chiral center is an atom, or more specifically, if I want to define it in terms of amino acids, I can say that a chiral center is an atom, or is a carbon atom, that has 4 different groups bonded to it in such a manner that it has a non-superimposable mirror image. Now, in all textbooks, the idea or the term chiral center has been replaced by chirality center, but the meaning and the definition remains the same. So each amino acid will have a chiral center. So if I look at a typical amino acid, it has a chiral center, and the presence of this chiral center results in a molecule that is different in the top, bottom, left, and right. So if we look at this particular structure of an amino acid, and this is a specific amino acid that we are looking at here, we looked at this amino acid previously, and this particular amino acid has a chiral center because the 4 hands of the carbon atom are bonded to 4 different chemical groups. This particular aspect is marked in red because this is the side chain of an amino acid. Now, these four chemical groups can occupy two unique spatial arrangements. Therefore, the amino acids have two possible stereoisomers which are non-superimposable mirror images. And therefore, this kind of isomerism is sometimes referred to as enantiomerism. As I told you, the details of stereoisomers and stereoisomerism will be dealt by Professor Bayumi in the section related to isomerism in the Foundational Concepts course. But what we need to know for amino acids is that all amino acids will have two possible stereoisomers which are non-superimposable mirror images. I told you till now that all amino acids will have two non-superimposable mirror images. But of the 20 amino acids that we need to study in this course, there is one amino acid that does not have two superimposable mirror images because that specific amino acid does not have a chiral center. So I wanted to explain this particular aspect using a specific question which you may encounter during the assessment. So this is a typical multiple choice question that you will encounter in a biochemistry assessment here at MBRU. So the question asks you, which of the following amino acid does not have a chiral center? Now, when I talk about, or when I mentioned the definition of chirality and chiral center, we talked about a specific aspect, which is that the carbon atom, in order to form a chiral center, should be bonded to four different chemical moieties. So if I want to answer this question, I need to look at the structure of the amino acids and then identify the amino acid that does not have four different carbon atoms, four different chemical moieties attached to the carbon atom. Or in other words, I can say that that specific amino acid will not have a chiral center. Now this is phenylalanine. Does it have a chiral center? The answer is yes, because the four chemical groups attached to the carbon atom are different. Threonine, yes. Leucine, yes. Lysine, yes. But when I look at the structure of glycine, I find that the four chemical moieties attached to the carbon atom are not different. Yes, one is carboxyl group, the other one is amino group, but look at here, you have two hydrogen bonded to two different hands of the carbon. That means this specific amino acid does not have a chiral center. So the answer for this is glycine. As you understand the structure of the amino acid, this particular exercise will become easy. So what you need to focus on is the structure of the amino acid. Sometimes students are asking, should I memorize the structure? The answer to this is that for assessments here, you don't need to memorize or remember the structure of the amino acid, because during the exam, the structure of the amino acid will be provided to you. Let us now summarize what I just said, glycine is the only amino acid of the 20 amino acids that we study in this specific course that does not have a chiral center. So what we need to remember here, all amino acids or all molecules with a chiral center are optically active. That means all amino acids except glycine are optically active because all amino acids except glycine have a chiral center. And when I talk about optical activity, and many of you may have studied this in high school, is that any chemical molecule that has a chiral center rotates plane polarized light. So all amino acids except glycine rotate plane polarized light. So let us try another MCQ here. Which of the following amino acids does not rotate plane polarized light? This is just a variation of the previous MCQ. If you do not remember which is the amino acid, which you don't need to remember, but you can work it out, what you need to look at is the amino acid that does not have 4 different chemical groups attached to the 4 different hands of the carbon atom. And the only only amino acid that has such a chemical signature is glycine. Because if I look at all the other amino acids, you can see here the carbon is bonded to 4 different chemical moieties. Only in glycine there are 2 hydrogens attached to 2 different hands of the carbon molecule, which makes glycine optically inactive. So glycine is the amino acid that will not rotate plane polarized light. As I told you, this is a variation of the MCQ that we solved earlier. In an exam, you don't need to memorize such kind of information, but if you can apply the concept of chirality center, you should be able to answer this specific question. We move on, and in this first slide set, we will focus on 2 structurally unique amino acids. And why we need to focus on these amino acids is that in an assessment, most of the questions revolve around the unique amino acids. Now, we have been talking about amino acids. Of the 20 amino acids, there is a specific amino acid that is an exception to the general structure of the amino acid. On this side of the slide, we can see the general structure of the amino acid. Again, recapping what we have learned so far, we have 4 different chemical groups attached to the 4 different hands of the carbon atom. This is the general structure. However, there is an amino acid which is called proline in the list of 20 amino acids that we need to study, where the amino group forms a ring with the side chain. So this is the amino group and this is the side chain shown here, and they form a ring structure. So incidentally, proline is not an amino acid, but sometimes some people like to say proline is an amino acid. This particular moiety that is formed here by the fusion of the amino group with the side chain is called the pyrrolidine ring. This is just for your information. I generally do not ask recall type of questions, but what is the ring that is present in proline? The answer to that is pyrrolidine ring. Let us try to frame an MCQ so that you can understand what kind of questions you may expect. The presence of the pyrrolidine ring will identify which of the following amino acids. You are given a list of 5 amino acids. You know that a pyrrolidine ring is a particular structure that is formed by the reaction of the amino group with the side chain, and such a reaction takes place only in proline. So if I give you 5 amino acids and I ask you which of the following amino acids has a pyrrolidine ring, the answer to that is proline. This particular group of the pyrrolidine ring is called an amino group, and therefore proline is the only amino acid that is an amino acid. Next, we come to the last part of this first set of slides, sulfur-containing amino acids. Generally, when we talk about amino acids, we have carbon, hydrogen, nitrogen, and oxygen present, but there are 2 amino acids that have sulfur present in them. And of course, this sulfur is present in the side chain. Okay, the sulfur is present in the side chain. The 2 amino acids, methionine and cysteine. Now, methionine is a sulfur containing amino acid, yes. In case of cysteine, this particular sulfur forms a chemical group which is known as the thiol group. I repeat, in cysteine, the sulfur present in the side chain forms a chemical group that is called the thiol group. A thiol group is chemically active. Actually, it has very high chemical activity. So that gives cysteine a unique chemical property. What will happen is that if we have 2 cysteines close by, the thiol groups will react with each other. So they will form a link between each other. I repeat, if we have 2 cysteine molecules close by, they will react with each other through their thiol groups, and they will form what is known as the disulfide bond. So there is a particular way to identify. Somebody says in a protein there is a cysteine. Cysteine means there are 2 cysteine molecules which are existing as disulfide bond. That means one cysteine will link with another cysteine. The thiol group is chemically active. In nature, in order to attain stability, the chemical group needs to be inactive. Therefore, the thiol group of 2 cysteine amino acids will react with each other and they will form a disulfide bond which is called the cysteine. So cysteine means there are 2 cysteine molecules. Will cysteine always be present in nature as a cysteine? The answer is yes. So and we will talk about this when we talk about protein structure, but I want to introduce you to the concept. So you have a protein in which you have one cysteine, and cysteine has a thiol group which is highly reactive. What it will do is it will form a disulfide bond with another protein molecule with another cysteine. So there will be two proteins linked to each other in such a case. Which is the amino acid that has a thiol group? The answer to this is cysteine. So that's the end of the first set of slides. Before we go, I just need to mention that during the formation of cysteine or disulfide bond, there is loss of hydrogen. That means the conversion of thiol to disulfide is an oxidation reaction. I can therefore summarize what I just said as a single sentence that cysteine is oxidized to cysteine. When I tell this, I'm talking about two cysteines reacting with each other via their thiol group. That brings me to the end of the first set of slides. The rest of the lecture on amino acids will be divided in other sets. Kindly follow and comment on the slides in the YouTube channel that you have been introduced to in the introductory session of enzymes and metabolism. Thank you very much.